Right.
This meeting is being recorded.
Okay. Yeah. All right. So welcome, Lucy, and welcome everyone to the Colloquium. Lucy Colwell is a faculty member at Cambridge University in the Department of Chemistry. She's also a research scientist at Google. Although she's a professor of chemistry, her BA is in mathematics from Cambridge. And she has a PhD from Harvard University in applied mathematics. Lucy did postdoctoral work at the Institute for Advanced Study in Princeton. She's a leader in the field of interpreting protein structure and function from amino acid sequences. Her work is highly interdisciplinary and uses tools ranging from deep learning to physically motivated models to understand enormous data sets of biological importance. Her contributions were recognized by a Simons Investigator Award in the mathematical modeling of living systems. So we are delighted to have you here to tell us about this exciting work. Welcome, and the floor is yours.
Well, that's great. Thank you. Thank you, Harsh, for such a kind introduction. I'm actually in this talk mainly going to talk about some of the recent deep learning work that I've been doing. And I've actually been doing that work in conjunction with a number of researchers at Google, which is why I'm here in Cambridge, Massachusetts, rather than in Cambridge, UK at the moment. This was just an exciting opportunity for me to be on secondment, is how we sort of say it in England, where I'm on leave from the university and I'm at Google at the moment. And basically there's just a lot of expertise at Google that isn't available to me at Cambridge. And so that was what made it sort of an exciting place to spend time. I'm really fascinated by some of the machine learning results that have come out and the sort of the abilities of these models to really understand systems from really seemingly very limited amounts of data. I think there are a lot of deep questions to be answered in that area as we learn more about the abilities of these models. Right now, I'm sort of in a very pragmatic phase of research where I'm really just trying to understand what the models can do, particularly in the area of making accurate predictions from protein sequences. And so this talk is sort of scarily devoid in some ways of deep science, I would say. So there are results that I'm really excited about, but there are not like deep theoretical explanations. Hopefully there will be, but you know, I'm still sort of trying to figure out the results. So this is, so we're really interested in the problem of predicting protein function from sequence. I worked a lot in the past, and many others have recently been working on the problem of structure prediction, which is of course fascinating, and a lot of exciting progress has been made. I was involved in some of that progress. We were able to solve this solve is a strong word, to make progress on this in an unsupervised manner by using information extracted from the correlation patterns of large sequence alignments, and then use kind of maximum entry approaches to fit models to that data. A really classical approach that's been popular, you know, obviously in physics for a long time, and we were able to apply some of that and use it to make progress on structure prediction. And some of that work has folded in to AlphaFold. Sorry, that's a terrible pun. It's been incorporated in AlphaFold in that they use some of these pairwise features that come from the maximum entry models as part of the input. And so that is really exciting. And being able to predict structure is great. But even with when we made this progress, which was about a decade ago, we could predict very nice structures if we had enough sequences. But that didn't actually get us all the way to be able to predict function, which I think is really sort of the end goal in many ways. You know, it's great to know that this sequence of amino acids results in this tin barrel, but the fact that it fluoresces green, and you know, this is of course GFP, has had enormous impact on countless areas of science. And telling that just from the structure, in particular telling whether it fluoresces green or blue or red or any of the other colors from the structure is extremely difficult. And being able to make predictions of how changing the sequence will change the function, I think is really an important problem. I'm very excited about the idea that we can really go straight from sequence to function without necessarily needing to be able to predict structure, although if we can, that obviously is also great. So we have access to lots and lots and lots of sequences. I think I have a figure here showing this is the growth of sequence data over the last few decades. It's about a year out of date now, but this exponential has not stopped going up. These are log scales on the axes, and it shows no sign of sort of plateauing. We have billions of sequences in the database that we have access to. And if we could only understand and exploit that information, it could be transformative in many areas of sort of human activity. And so being able to exploit this wealth of data to discover new proteins, new functional activities, I think has the potential for really significant impact. But many of these sequences, we still don't actually know what they do. And that's a really significant problem. I mean, for even more of them, we don't know what their structure is, but just having any idea at all what these sequences do, something like a third of microbial sequences, we simply can't annotate. They are in the database as uncharacterized protein, and we know nothing about them. That seems a really sort of troubling situation from my perspective. So an approach that's received a lot of attention recently and won the Nobel Prize in 2018 is directed evolution, which is a way of finding better versions of sequences. And this is essentially involves random local search and selection. So here you're starting from a sequence where you know what it does, and you're making random changes to it and asking, does that improve its activity? So this is, a state-of-the-art technique, but it's still very challenging to be able to take a sequence, a starting point, and engineer it to obtain the activity that you're looking for. And I see these two problems that I presented very quickly as being very closely related. So being able to predict the function of a completely new unknown sequence, or being able to change a sequence to optimize its activity, they all involve being able to understand this relationship between protein sequence and functional activity. And of course, there's many relationships involved there, right? Because there are lots of different functional activities. So this is sort of a many-faceted problem. But why is it the case that finding new proteins is so hard? And we've all unfortunately seen recently the issues with searching for antibodies. When you're infected with any kind of pathogen, your immune system carries out a search for antibodies in order to defend you from that pathogen. And a typical antibody has at least 30 variable sequence positions. I'm thinking of the CDR, the floppy loops of the antibody that dictate its binding specificity. So if there's 30 variable sequence positions and there's 20 amino acids, and that right away tells us there's a really very large number of possible antibodies. In your body at any given time, there's something like 2 to the 38 unique antibodies floating around. So that also is a very large number, but it's nowhere near as large as this 2 to the 130. And so this is why it's hard for people to defend themselves against pathogens. And so my pitch here is that we can really leverage some of the advances in machine learning in order to capitalize on this large amount of natural sequence diversity that we have in databases, and then run sort of active learning loops where we build models that predict the activity of unseen sequences, and then test them in wet lab experiments and sort of run this loop. So I'm basically imagining a case where we put machine learning or modeling into a directed evolution type workflow. So here, we have this case where you actually make specific sequences, you then test them in the laboratory, use that data to build a model that relates sequence or some kind of representation of sequence to functional activity, and then use the model to choose the next set of sequences that you design and put into your assay. There's a sort of nice cycle that you can go around in order to really actually learn this relationship between sequence and activity, and thereby walk through the space. So here, there's an optimization step where you actually query the model in order to identify the best sequences, and thereby find sequences which, in this case, actually, this is a non-local optima. So this is, you'd have to sort of essentially cross a valley. This would be the local peak that you'd reach with directed evolution. And the idea is that by using machine learning, you'd be able to find this distant peak. This is obviously a sort of cheesy three-dimensional representation of a very high-dimensional space, but the idea, I think, hopefully holds true for that high-dimensional space, that by building accurate models, we can really find better sequences than are accessible using current techniques. So there are a few different topics that I want to touch on in the next few minutes. I'm first going to talk about a set of work that actually was finally published just last week, where we're using deep models to learn representations, and in the process we're actually annotating or demonstrating that we can annotate protein sequences more accurately without using alignment. I'm then going to talk about some work where we use sort of in silico objectives in order to develop and compare model-based optimization strategies. That's strategies for taking the model and walking through sequence space. We also have some work using or developing strategies for large batch optimization. So if we're testing a batch of sequences, how should we divide that up? And finally, we have some nice experimental validation results, and hopefully I'll be able to spend most of the time talking about that, because in silico results are all very nice, but it's only really when we test things in the lab that we know whether they really work or not. So the first section, where I'm going to talk about being able to annotate some of the diversity of uncharacterized sequences that are currently in the database, was really motivated by this 2018 paper, where the abstract started with this claim that 1/3 of all protein coding genes from bacterial genomes simply can't be annotated with a function. And this struck me as, I don't know, really just an atrocious state of affairs, something that we should try and fix. And in this paper, they were proposing a very sort of laborious experimental procedure that allowed them to look at 12,000 of these genes, but there are billions of protein coding genes in the database, and so we really need computational tools to enable this to scale. And the existing computational tools have been extremely successful, and yet somehow we still can't annotate a really large fraction of the proteins we have access to. And so we decided to start off by working with a database called PFAM, which essentially is built using a set of hidden Markov models. So there's a sort of small set of sequences that are human verified and sort of chosen and curated very carefully by humans. And they're used to fit about 18,000 models, and those models are then used to build the large database. So it's this sort of small seed part of the database that we were interested in because it had sort of human curation, and we felt that we wouldn't just be trying to reproduce a different computational annotation tool. And so basically, there are 17,929 classes, so just under 18,000. The sequences vary hugely in length. There are very, very, very many really small families that are shown right on the left-hand side of this plot here. There's almost, there's thousands basically of families that have less than 25 sequences. So this is not a canonical deep learning problem, in that there isn't huge amounts of data. There are some large families, but not comparatively not that many. And so we built, this is essentially a ResNet. So this is a pre-existing type of deep neural network that was used with a lot of success on images. We used it for sequences. So we're using one-dimensional convolutions rather than two-dimensional convolutions. And essentially, we're just putting in unaligned raw amino acid sequences into this model. And then we're training the model so it learns a nice representation of every sequence. And amazingly, no matter how long the input sequence is, the representation is 1100 dimensional, and it's the same for every sequence. And so we learn this nice representation, then use it to make predictions. And it really turns out that proximity and that learned representation tells you about the family that a sequence belongs to. And so, it wasn't like that easy to train these models and get them to work, but we were fortunate that this nice architecture already existed that we could just sort of make use of for this problem. And so this figure shows the performance of different models. Our models are the ones, there's a prot CNN, which is the CNN model I showed you just now, and there's a prot ENN, which is an ensemble of those CNNs. Essentially what we're showing here is error rate. So here we want the error rate to be low, and it's shown on a log scale simply because the models essentially all do very, very well. You can see these errors are quite small, and so to sort of separate out the curves, it's easier on a log scale. And I've stratified the performance by the distance of each sequence, each test sequence from the training data. So we made a test train split of the data. It was a random split. And of course, when you make a random split, you have to be very careful because some of your test sequences can be very, very close to your trained sequences. And so we stratify performance by, this is the distance from each test sequence from the training set. And the reason it says average here is it's the average within each bin. So we have different bins, and I'm just showing you the points of the average error in that bin. And essentially, the existing models involve HMMs, or BLAST is probably an approach that everybody has heard of. These all involve alignment, and they're making significantly more errors overall than our deep neural networks. And we're also excited that the deep neural network was able to really perform well, even really when the sequences were really far away from the training data. So that was exciting. We also made a clustered split where we clustered the data before and used the clustering to guide the split. So we wanted to make really sure that we weren't reporting results for sequences that were identical to the training data. This is a little bit technical, but we made this clustered split, and we had the same result there, that again, the error rate is lowest for our ensemble of neural networks. It does get really hard as you get really far away from the training data, but the neural networks are still able to do better. So this was exciting, but what was even more exciting for us was that these methods really provide complementary information. So here we're showing the HMM in this sort of ochre color, and then the neural network is in blue. So the neural network does slightly better than the HMM, but together actually they do even better. And that tells us that they're really learning different sort of things, different patterns on with which to classify the sequences. And so we really focus on this ensemble as being the way to kind of move forward. And using that ensemble, we were actually able to add 6.8 million annotations to the database. And among those 6.8 million sequences, there were 360 human reference proteome sequences, so 360 human proteins that were previously uncharacterized that we were able to annotate using this approach. So this was very exciting. We released this earlier, I think about a year ago, actually, March last year with PFAM, and the paper just came out last week. So we're able to make something like a decade's worth of progress in terms of improving the annotations in the database. That was really exciting for us. We've also done some work where we've been using language models essentially in order to try and push the performance even further. So here we're pre-training a transformer model. So a transformer model is a model that sort of uses attention and sort of looks at similarities between different places in their sequence. We're pre-training this model across millions of sequences, in this case from a database called UniRes50, and then we're taking that pre-trained model, kind of freezing all the weights, and just using the very top of the model, retraining that to do our classification task. And so this is really based on exactly the objective that's used in language modeling, where essentially what you do here with your training data is you mask out some of the characters, and then you ask the model to predict them back. And in that way, you're sort of able to really increase the amount of training data you have, because you don't have to have labels for each sequence. You're just literally deleting part of the sequence and predicting it back. And so, we picked something like 15% of the input tokens at random, and some of those got replaced by a mask. And so then essentially that just training the model on that task gave it enough knowledge about sequences to kind of warm start it for the classification task. And so it turned out that when we do that, we found that larger models do significantly better at that task of predicting missing amino acids. So here is a model with 1 layer compared to a model with 12 layers, and the 12-layer model does quite a bit better, as you can see from this accuracy statistic. And then we did find that actually translated into better performance at our classification task. So here are, this is the parameter count on the left-hand side, and here are the performances of various different models. I showed you prot-CNN and prot-ENN previously, and this is our new transformer-based model that has been fine-tuned. And you can see that it's actually doing 1.5 points better at this cluttered split. And what was exciting for us is it actually has fewer parameters than the ensemble we were working with before, which means that inference is quite a bit cheaper in terms of compute. And it means it's actually practical to serve that model interactively, which just makes it easier for everybody to use. So that sort of pre-training gave the model additional power for sort of being smaller. And yeah, we can see here I'm actually showing accuracy as a function of distance from the training data, and you can see that the purple is the new line I've added, and it's sort of doing quite a bit better than the existing ensemble. And the HMM, is actually competitive, but it's just for these really distant sequences that we're able to do better. What surprises me is that the HMM actually has a lot of parameters compared to our language model. So I hadn't expected those to be comparable numbers, but they were. I'll skip over and go on to talk about optimization strategies. So essentially, these deep models we use to learn like a nice representation of sequence space that we could use to build our models. But of course, once we have that model, in order to sort of work with this loop, we need to be able to actually move in the space and optimize the model. And that raises a question, because amino acids are discrete, we can't sort of just follow the gradients, because we're going to end up sort of with a half amino acid or a fraction of amino acid. So this is a sort of interesting discrete optimization problem. And so we have to be able to solve this problem. And discrete optimization is sort of an interesting activity, it turns out. And so, yeah, solving this optimization problem is what I'll talk about for the next few minutes. And in order to make progress on this, we really had to turn to in silico benchmarking, just because running lab experiments is expensive and time consuming, and really takes a lot of people's time. And so to be able to try lots of different things, it was essential to have like computational oracles. When we talk about, these software-defined optimization problems, the idea is they mimic wet lab design tasks. They're never going to be perfect, right? Because we don't have protein fitness landscapes in the computer. If we did, that it would be really nice, but we don't understand them well enough to be able to build models. So we're trying to sort of mimic the features of the true optimization landscape, and it allows us to ask questions about, how should we budget our experiments? Should we use initial labeled data, and what kind of metrics are useful? And we can make problems with different interpretable levels of hardness. And so we're attempting model-based optimization here, where essentially, there's a real experiment that we want to do, but the evaluation is very expensive. And so we want to sort of label a small number of points and then fit a surrogate model on those points, which is cheap to evaluate. And then we define like an acquisition function over that surrogate model, where they're sort of balancing the trade-off between exploit and exploration. Like, how much should we, sort of exploit the model we already have and take, test the sequences it says are best, versus how much should we try and test new sequences in order to improve our model. So that's sort of that trade-off is defined or captured by this acquisition function, and then we optimize the acquisition function using what could be a sample inefficient method like reinforcement learning or evolution or cross entropy, because this is cheap to evaluate, so we can just do this in silico. And then once we found the best sequences, that the sequences that optimize the acquisition function, we can actually go back and test those in the lab. So this is where we're trying to get to eventually, and we're trying to figure out right now. We're going to isolate this piece here, this optimization piece, and use sample inefficient methods to explore it. So we really want to make this whole approach more robust. Essentially, when we're sort of running this model-based optimization loop, we're going to have situations where we have only a small amount of data to start with to fit our surrogate model, and so therefore in that point we expect the model to be relatively unreliable. And then as we sort of collect more data by going around the loop a few times, we might have a better model, so we might be able to trust it a bit more. And so that means that the model complexity should adapt essentially to the amount of data we have to fit the model. And so we implemented like a sort of automatic model selection approach that basically split the data and selected the best model based on performance on held out test data. We decided, we only want to use the model if it's accurate enough. And also these models tend to have really vivid imaginations. So, you give it some data, which is of course in some small area of sequence space, and the model fits that data and then has the idea that if you just move a bit further away, like suddenly the best sequences are going to be found, or really great sequences will be found. So the models become quite unreliable as you extrapolate, at least in some cases. And so we found that we had to really enforce some kind of trust region in order to stop the models being overly optimistic. And we've actually found that this is an example where for our in silico oracles, this has been a real problem, whereas in the lab actually we've found that we're able to extrapolate with great success. And so that's somewhere where the sort of gap between simulation and reality is wider in opposite direction than we might expect. And so we also found that the error of the model and the uncertainty are pretty closely correlated. Of course, we can measure the uncertainty as we go, but we don't know the model when we're using this prospectively, but this is a useful proxy. And so we can simply measure the uncertainty of the model as we go, and then threshold that. And once the model becomes too uncertain, we can stop training and sort of go back and collect more data. Essentially, it's a signal that we need to do more experiments. So this is sort of the inner round that we're on, and then at some point we need to switch back to the outer round and collect more data, and then go back and do more inner loops. So we built a number of, these are different in silico problems. They're based on particular real data sets, essentially. And what we're showing here is, as a function on the x-axis, is the number of samples, so the number of experimental data points we have to collect, and the y-axis shows the maximum seen so far, the sort of best example that's been found so far in this optimization. And you can see it makes quite a difference what approach you use. The random approach takes a very long time and doesn't actually find the best sequence on the sort of scales of samples that we're looking at. Whereas in contrast, some of these more sophisticated approaches are able to rapidly find good sequences. This paper was introducing this DINA PPO approach, which was a model-based optimization that used a particular reinforcement learning policy. I'm not going to go into the details just because of lack of time, but essentially we found that there were situations when reinforcement learning really provided good performance across a range of these problems. However, the reason I'll talk about large batch optimization next is that there were also cases where it didn't work that well. So we have cases where this model-based optimization using that sort of reinforcement learning approach really did well. There were places where it really didn't perform well at all, and other methods were able to do better. So this pointed to a lack of robustness with our approaches. So what we realized is that when we're testing sequences in the lab in this outer loop, essentially you always test many sequences in parallel simply because it's cost effective to do this, and so we can exploit this parallelization in order to get better data. And so the question sort of becomes, how should we split up the different batches of sequences? If we just have one algorithm, we're going to use the same algorithm to design all of every batch, which is shown here in green. But of course, what we could do instead is have multiple different algorithms and assign them different proportions of each batch according to how well they're performing. And so essentially that allows us to kind of exchange information between the different methods, because we're fitting each method on all the data, so the data collected by all methods, but then we can divide the batches up and sort of take advantage of having different approaches. And it turns out that if we use an approach which we called P3BO, so this is population-based black box optimization, then it consistently does quite a bit better than the other approaches. And this is a rank max reward. This is a ranking where 6 is the best and 1 is the worst, which is counterintuitive to me because I think 1 should be the best, but nonetheless, in general, this P3BO population-based ensembling method is able to outperform the other methods. You can sort of think about it as being at least as good as any of the other methods in general. I mean, that's not quite true. That there are edge cases where another method could do better, but it should overall basically be at least as good. And so, yeah, here is one of those edge cases where one of the other methods is able to do slightly better. But essentially, the ensemble method should be able to do pretty much as well as the best population member. And then you can sort of take that even further, where you start not only changing, sort of weighting the proportion of each batch by the amount, by the performance of each algorithm, but you can also mutate the hyperparameters of each algorithm, and this kind of becomes a bit insane essentially. But you can eke out like a little bit more performance by pushing that even a bit further to actually change the algorithms themselves. Perhaps we got a little bit too obsessed with this problem in some sense. But what we did find was that by ensembling across these different methods, we could actually really increase the diversity of sequences that our models or our methods were able to design. And that's important because, of course, for sort of drug discovery tasks or other problems, often you find like a lead candidate or a best sequence, and it turns out that it fails for some other reason, like it has some sort of horrible toxic effect, or it aggregates in solution after a short amount of time, so it's not practical as a product. And so having a diversity of different solutions is a real benefit. And so here these points are colored by the algorithm that generated them, and you can sort of see that the points tend to cluster to some extent, except for the top few algorithms that are sort of spread throughout the space. Okay, so I've talked a lot about computational work and sort of in silico work. I'm now going to talk about experimental validation. I'm going to spend some time talking about a case study where we worked with some collaborators working on gene therapy. And then if we have time at the end, I'll switch and talk briefly about a peptide design challenge. So this is a collaboration with a group at Harvard in the Wyss Institute. They were interested in AAV-based gene therapy. So AAV stands for adeno-associated virus. This is a virus that we all encounter every day. It's very common. We all have antibodies against it. There's a whole approach to gene therapy that uses AAV capsids. So essentially, you take the capsid of AAV and you encapsulate your gene of interest inside the virus. And essentially, this capsid is important because this is what your immune system sees when you receive the therapy. And because we all encounter AAV every day, we all have antibodies against it. And so that means that our bodies would respond by breaking down the therapy. Antibodies would attack it, and that's not much use because it means that we don't get the effect of the therapy. And so it's really very important to be able to design diverse new capsid proteins. We need to be able to change the surface of this protein so it's no longer recognized by our antibodies. Ideally, it would also be addressable so that you could send the therapy to the tissue of choice and therefore have to give people much lower doses, therefore avoiding potentially sort of toxic effects. So those are two very important goals for the field. In order to reach these goals, being able to design diverse variants of this capsid that are able to assemble into that beautiful symmetric object, and also package their own genome. So this is a packaging phenotype. This is like an important, this is necessary but not sufficient, I guess, for both of these other phenotypes. And so that's the phenotype that we focused on. So we want to be able to design massive, very different versions of this capsid that still function at being able to assemble capsids and produce progeny, essentially. So assemble functional capsids that contain copies of their genome. So this is the map of the genome. This cap gene is essentially one of the two main genes inside AAV. There's the rep gene and the cap gene, and then some other sort of pieces. And essentially, this is the capsid is formed from 60 copies of a single monomer, which is about 735 amino acids. So it's not a small protein. And yeah, it's this beautifully highly symmetric object, which is about 25 nanometers across. So this is not an easy design target. We're going to make changes in this protein and ask that it still, assembles into this capsid. It has to be integral, so it can't have any holes, and it also has to package its own genome. And so we focused on a small region of this protein as a sort of proof of principle. And we started off by making all single mutants and also sets of randomly chosen multimutants. So we just made changes at random, which sort of, sounded kind of nuts at the time and was kind of nuts. And there's a complicated experimental process involved. But essentially, for every sequence that we design or that we put into the assay, we get a measure of its performance, which is like a selection coefficient. And we compute that by sequencing the initial library and the library after a round of selection. So we ask that the viruses survive a round of selection and that their DNA is contained in integral capsids after the round of viral replication. And so then we sequence the final library, and for every variant we have the number of copies at the start and the number of copies at the end, and we simply compute a ratio. We typically take a log of that too, just to kind of make the numbers sensible. So we're working on a small region of this very large capsid. This is shown in context in the 3D structure. So some of this piece of capsid is very buried and so harder to change. Some of it is more surface exposed and so potentially easier to make changes to. And of course, there's 60 copies of this region in any given capsid. So if we change it, we're changing many places in the capsid. And so, it really has to still function. There is some diversity among natural sequences, and in the sort of part of the region that's more surface exposed, there's a bit more diversity. This is showing entropy. So across 238 sequences, which amino acids are used. Some positions are completely conserved, and those are shown by these very tall amino acids. And some positions change quite a bit. So we made all single mutations. So this is showing the positions in that region. It's a 28 amino acid region. This is showing all the amino acids. And essentially the changes that break the capsid, the single change that breaks the capsid, are shown in blue, whereas the single changes that don't break it are shown in white. And you can't really be much better. So red would be that you were much better than wild type, but at packaging, you're not really going to get much better, essentially. So you can see some sort of slight improvements perhaps, but essentially the colors here are either white or blue. We use this single mutation data to make an additive baseline model. So we're going to design multi-mutants by simply combining mutations with good single site outcomes. We're just going to say that they add. And then we can use that very simple model to walk in the landscape. And so we can simply, take the best, we can make a single algorithm where we take the very best single mutations and combine them and keep going until, we decide that, we've added too many mutations, or we had an approach for doing this. And we use that to design 56,000 sequences, all of which, should have been good. So when we looked at the data that we get out of sequencing, it's quite noisy. And so we ended up deciding that there was basically one bit of information in this data. So this was for a set of controls that were either viable or non-viable. You can see that there isn't much overlap. So it is the case that we can probably separate reliably between viable and non-viable sequences. But this is the data from the whole of our 56,000 sequence library. And you can see it sort of basically overlays our controls. And so we decided that basically we could say whether each variant was viable or not. And so I'm plotting the results for the 56,000 here on this chart, where I'm showing distance from the wild type or starting sequence on the x-axis. I'm showing precision, the proportion that were accurate, that worked essentially on the y-axis. So you can see that if we just make 2 changes, actually most of the capsid still worked perfectly well. But once we're making sort of 7 to 10 changes, or even 11 to 20, it was much harder, and the precision has really dropped off. So I was actually surprised even by this initial result that we can make changes at random and the capsid will still work. So the space is surprisingly plastic. What we wanted to understand is whether we could learn better models to design multi-mutant sequences. Can we learn models that would make this curve kind of go up and improve precision? So we used 3 different architectures. We had a sort of logistic regression model, which is a baseline approach that doesn't really capture interactions between amino acids. Then we made a convolutional neural network and a recurrent neural network, which had many more parameters than the logistic model. And we used ensembles. So we took that inspiration from our theoretical work to make ensembles of each type. We didn't ensemble across all 3, which might well have been a better approach. And so we wanted to test how well these models performed on the data that we already had. So we had the 56,000 design sequences I told you about, and also a set of random multi-mutants that we collected data for. And so this is the additive data was much more sort of positive. We used that as held out test data, and then we used these randomly designed sequences as training data. And we constructed sets that either contained just 1 or 2 mutations, or we used all of this data in order to train models. And in that exercise, we found that actually, yes, we could. So even if we're just training on these sequences of 1 or 2 changes, we actually can extrapolate quite well. And using the neural networks in particular, we can make more accurate predictions as we get sort of 7 to 20 steps away from the starting sequence. So the models can extrapolate, even given a very small amount of training data, which was somewhat surprising. So that was a retrospective validation. Of course, we needed to test this prospectively. And so for that, we made 3 different training sets. So the 2 that I told you about, and we also just put all the data in to make a 3rd really large training set. And we had our 3 different model types. And so we took the cross product of these to make 9 total models. And then we used those trained models to kind of screen and evolve large sets of candidate sequences. So, we can't screen all the sequences because the space is really very, very large. So instead, we picked a couple of billion random sequences at different distances from our wild type starting point, which is shown in pink here on this diagram. Basically, we just took sets of 100,000 sequences at different distances between something like 5 and 29 steps from wild type, and we used our models to score all of those sequences. And so then once we'd scored all those sequences, we also used the models to evolve the sequences using a simple approach where we simply flip amino acids and score them and keep the ones that are good, essentially. So we use this more guided design for 20 steps. And so we ended up testing a lot of sequences. So there's like a lot of details here, and the choices are important, but I don't know, it's not particularly exciting, I wouldn't say. But we ended up with some quite nice results. So we're training on, just a bit less than 3,000 sequences that either had 1 or 2 mutations, and then we're asking the models to design sequences that are up to 29 mutations from the starting point. So we're really asking them to extrapolate. What we found was that our logistic regression model sort of fell over and really didn't do well at all, which was quite surprising, because given a small amount of training data, you'd expect a simple model to do well. So we don't really understand what happened there. This is our additive baseline that I talked about before, and you can see that it sort of really drops off as you get further away from wild type. But the CNN and the RNN are able to accurately design sequences quite far away from the starting point. So they extrapolate really quite well. And this was very surprising to us. So, here at 18 steps from wild type, we're still able to design, well over 1,000 viable sequences using the RNN, which is something like 50% of the candidates that we put forward. And that was just, an extremely surprising result. We published this last year. And what we found was that, that small training set, the performance there actually, mirrored the performance with larger training sets, except that curiously the logistic regression model was able to do extremely well with this larger training set. It basically seems as if the logistic regression model is able, in this case, to identify like a ridge in the fitness space and walk along it. It's really extremely accurate, and that was very exciting. And then with the really large training set, we sort of found that somehow the neural network models really were able to take advantage of that large amount of data. And the logistic regression model did okay, but it didn't quite repeat its performance with the medium-sized training data set. So I think it was exciting that even simple models were able to do really well at this. One thing I'll go into later is the diversity of sequences designed, and so there were some trade-offs we found there. This is just looking at what mutations were viable, and so the additive model, the random mutations in the additive model, we made both substitutions and we also made insertions, and the additive model kind of struggled in this first part of the tile, which was more buried. So it was able to make some substitutions, but basically no insertions. Whereas in contrast, the logistic regression model was able to make some insertions in this first part of the tile, but the neural networks were able to kind of amplify that and make mutations, particularly also everywhere in the later part of the tile, but also more frequently in the earlier part of the tile. And we also saw that with the RNN. So the networks were able to kind of explore in a way that the simpler models weren't. We used sort of projection methods to try and look at the sort of diversity or the amount of sequence space that each model covered. So this is just looking at the training data. If we look at the walked sequences for each model, we see that the logistic regression walks are sort of more confined in sequence space than the neural network walks, but it's really very difficult to judge scale using these projections. And so we made a simple approach to quantifying diversity, where we just clustered sequences and used a kind of greedy agglomerative clustering approach to count the number of clusters, so kind of how many tiles, how much of sequence space each method was able to cover. And so there we found that there really was quite a much difference. So this is showing as a function of the cluster radius, or the size of the tiles, how many clusters of viable sequences each method produced. And so we see that the logistic regression model was really quite a lot less diverse than the neural networks, or indeed than the baseline, although they do cross over as we make the clusters larger and larger. So in some sense, this provides a measure of the area of sequence space in which each model discovered viable sequences, and we see the neural networks are really able to discover diverse viable sequences. And we're still trying to understand exactly how that works, and we sort of saw somewhat similar pictures at these different using these different training sets. And so I think an interesting point here was that having more training data didn't seem to add that much. So it seemed as if the model learned everything they needed to learn from sort of singles and doubles, which perhaps says something about the complexity of the space and the complexity of the interactions that we see in this space. So at this point, I have another experimental example I could talk about, or I'd be more than happy to take questions. I don't know what is most typical for this forum.
I'd like to ask a couple of questions.
Cool.
One, are you familiar with the concept of penetrance in genetics?
I'm not sure actually. Yeah, can you explain?
Well, yeah. So there was just a study published from I think it was a group in New York where they used the UK Biobank and another giant database sequenced human beings and found that the average penetrance, and I'll explain it in a second, of pathogenic variants was 7%. So what that means is that 93% of the people who have a particular variant will not express a disease associated with that variant. This is all by way of telling you that function, as defined biologically, is not just about the sequence of a protein. It's about the sequence of that protein in the particular context in which it finds itself. And that includes not only environmental variables, but also allelic differences at each of the other 20,000 loci.
Oh, yeah, no, I couldn't agree more. I think that if we're trying to go, this is the problem, of course, with GWAS studies, right? And trying to understand the functional effect mutations in the human genome. This is an immensely complicated problem. I would love to kind of be able to work on this problem, but as you point out, gene it occurs in isolation, in an organism, and the compensation that happens between genes, or between two genes or many genes, is enormous. And being able to understand this combinatorics is just mind blowing. That is such an exciting research problem, and I sort of see our sequence to function problem of looking at individual proteins, particularly for sort of biotechnology applications where they're kind of taken in isolation outside of their natural context. I see that as a microcosm of this much larger problem that you're talking about, which I think is immensely exciting and difficult.
And then with respect to the adeno-associated virus work, when you say a given variant is viable, is that taking into account one of the, and again, another point would be that most proteins are multifunctional, but are you taking into account the ability of the capsid to bind to the cellular receptor?
So the assay that we're working in for packaging only doesn't take that into account.
Because that's critical.
Absolutely, that is absolutely critical.
The related points in terms of the reality of using AAV as a vector would be that even if you escape the existing antibody, making all these mutations will probably result in the induction of other antibodies and T-cell responses.
Yeah, absolutely.
And the other problem you could run into is you may inadvertently create an epitope that is similar to a self-epitope that leads to autoimmune disease.
Okay.
So the latter point, I think that mimicking self is extremely difficult, but I don't understand enough about self versus non-self to be able to address it. But your former point about the fact that you're going to induce an antibody response is absolutely correct. And so this is why being able to sort of design diverse capsids, you'd have to give people different therapies essentially every time they needed a new dose, which is sort of impractical. And I think that better solutions will be found. But at the moment, our collaborators have actually started a company in this area, and I won't pretend to be the expert on this in any sense, but they've actually started this small company which has now signed deals with a number of big pharma players, and so it seems as if this technology is actually going to get out there into the real world, and they are absolutely have to address all of these problems. But I think being able to sort of move in this space has been very important to the success so far of their company. And so, yeah, I think that gene therapy is clearly, an area that has a lot of potential issues. I would be nervous to sort of make a company that does this myself, but I am extremely impressed by our collaborators who have really taken this all the way. And I think they now have 50 people or something in the company, and it's growing rapidly. And I very much hope they manage to succeed. But I think that the points that you're raising are completely, accurate, and sort of, yeah. What's the right way of putting this? I think you have to be brave to really do this.
Okay.
In that particular, yeah.
Thanks.
Okay, so I think we've definitely jumped into the Q&A. We were discussing, you were asking whether we should do Q&A now. I propose the following: that let's have in the formal session, maybe we'll ask a few questions and then we'll stop recording, and then we'll have more informal discussion. And at that time, if there's an opportunity, you can tell us about those additional results, or maybe the discussion will just take over. All right. So, yeah. With that said, I'm going to invite more questions. You can either put it in the chat or use reactions to raise your hand, and especially encourage the students to come in.
I have a question. I was giving students some time. Very interesting talk, Lucy. I really enjoyed it. I guess I wanted to ask a bigger picture type of question, since AlphaFold and the machine learning application to protein structure was deemed as solved and the like. Where do you see this going beyond just the static structure and making new neural networks and getting better and better matches to static structure to, I guess, a bigger picture question of the dynamics and going from, okay, we have this starting structure, but what are the dynamics of that protein moving to the function?
Yeah, I actually think that's a really interesting question. So for me, AlphaFold sort of solves some problem that maybe is kind of interesting, but I don't know, we have like 200,000 structures in the PDB anyway, right? I kind of feel like we know a lot about structure. But I think dynamics is really interesting. I think the big problem is that we don't have enough data. I think if we had more data, then we could actually use that data to constrain neural networks, and potentially sort of really blow MD out of the water and really do a better job, moving beyond sort of the approaches that we know so well with classical force fields. I think the problem is that so many of these parameters in these force fields are just not fit accurately, and we can't, right? That's nobody's fault, and we really can't do a better job. But I think some of the advances that have come about in terms of being able to differentiate through the problem. So if we had enough data, then we can make guesses about the parameters, push that through the simulation and differentiate that to update the parameters and work right now in cryo-EM in particular, which enables protein motion to kind of be discerned from the snapshot of many different conformations that you get in a cryo-EM grid, and sort of you get this snapshot of the different sort of motions that are being sampled, and being able to, it seems like we should be able to relate that to the energy landscape, and if we could sort of use that as a source of motion data, or to complement existing sources of motion data, in order to try and get at this problem. I think that would be really exciting. So what I'm trying to say is I think there's a huge potential essentially for using machine learning to make progress on this problem, which I think is an enormously important problem. And obviously there's sort of NMR data that you can put into that as well, and sort of, yeah, even other sources of protein motion data that are hard to come across. I think also there are lots of people right now trying to train neural networks to kind of speed up MD. So you sort of take the MD as ground truth, and then you use a neural network to try and sort of accelerate, which is, I think, an important approach too, right? Like MD it takes a lot of compute, and if we can do it faster, that would be good. But I think we have to really fix the sort of fundamental limitations of the MD. And I'm not an expert in this, but that's my sort of take on the situation.
Yeah, I think you bring up a lot of good points, and bringing up cryo and NMR. And I work in fluorescence, and Yeah, I understand getting experimental data and enough dynamics is hard in and of itself, but also with the different experimental techniques of getting the different, like a common data type of format that you could then feed into a network that spans those different fields would be important. And I don't think that coordination is there yet. So thanks.
Yeah, that is a really good point. Like being able to, one of the nice things about the structure problem, right, is you have the PDB with all that data so sitting there in a nice format, and yeah, kind of tackling that for other data types, I think is really important. Yeah, thanks.
More questions? I have one. Actually, I have two. I mean, I'm trying to think of a way of asking it as though it were a two-parter, but it's really two separate questions. So one is about the first part of your talk, where you pointed out, started with that review article and pointed out that, if we just count on experiments, it'll never happen. So you need computational methods. But is there some way in which your methods, and you also demonstrated that your methods were, competitive, is there some sense in which they're also higher throughput than other existing computational methods?
Yeah, we actually spend a lot of time trying to do performance comparisons, and this is very delicate because, of course, the exact machine that you're comparing on becomes extremely important, and also some methods are better suited to particular architectures, CPUs versus GPUs compared to others. But to make those plots for BLAST actually took us an inordinate amount of time. Like it just takes a really long time because you're doing all these little pairwise alignments essentially. And so I think it took something like a month for us to make one of the figures in our paper for BLAST. And I feel, yeah, the amount of compute involved was really large. The methods using neural networks can annotate something like 500 sequences per second, which takes advantage of being able to parallelize on GPUs, but is extremely fast. And so I think that does make a difference. Right now, we work with the people at EMBL, EBI, just outside Cambridge in England. They have a metagenomic database that has 2.8 billion sequences in it, and they cannot annotate that with existing methods because of the compute costs and the compute time involved. And so, we hope to release annotations for that with them in the next couple of months. And so there it really will make kind of the difference between being able to have annotations and not, which is satisfying.
All right, well, I think maybe what we should do is switch over to the more informal session at this point. And so let's thank the speaker. And I'm afraid we can't really make our appreciation felt adequately on Zoom, but thank you. That was a really exciting talk. And I'm going to hit